I'm going over it very carefully so we understand how adaptive immunity works. In terms of your final, you do have another test coming up. I got to figure out when it's going to be. But essentially, starting with this chapter down to the end, you have a test on that. And then you have a final exam that's comprehensive. So over everything, it's going to have 100 questions, multiple choice. Now, the final exam will sort of be like what you just did these past 2 days. Because the final exam is going to have 100 questions, we have to do it in 2 separate days. So you get one half the final one day, and then the second half on the other day. Total. No, 100 questions total. Okie dokie. So real quick, if we get to the slide I want to get to today, then I have plenty of time within class to go out and run your scantrons off and figure out what you got on your test. Yes, Faye?
Are we going to have a final review online for the entire thing?
Not really, because really the final review will be all the other review sheets put together. So in other words, I'm not going to test you on anything new. I'm going to test you on what you've been tested on before. If that makes sense. In other words, I'm not going to make a brand new review sheet because you've already had all of them up to that point. It's comprehensive based on what I've taught you. So I'm not going to make a new review sheet. You just go back to chapters 1 through, I think, 27. You already have all the review sheets and go back over what you should have learned. So that's the best thing I can do. Okay, so you already have it. Okay, real quick. So far you've just been tested on what? Innate immunity, basically. That's your first and second line of defense. Now, if they don't work, right, if they fail, then you go to the backup plan of all backup plans. You go to adaptive immunity. This is your third line of defense. Basically, this is where the specifics come in. So in adaptive immunity, it is very tailored. It is very specific to a particular, not even pathogen. We go beyond pathogen, okay? It is very specific to a shape, bless you, a shape that's actually found on the pathogen. So you don't even need the whole pathogen. You just need something from the pathogen to trigger it off. Makes sense? So it's that specific, just a shape. So keep that in mind. A shape is all you need to trigger adaptive immunity. And it's also adaptive because it also will recognize that shape. Then it will remember that shape so that when it sees that shape again, the amount of attack will be more quick the next time you're exposed to it. Makes sense? This is why you get vaccinations, right? Now, in terms of the 5 attributes of adaptive immunity, right, it gets better over time, right? It gets better over time. Every time you see that shape, it gets better and better and better, okay? It is specific, right? So I already mentioned that. It's specific in terms of it recognizes a particular shape on a pathogen or even a product from the pathogen. Say like a toxin, for example. That's enough to trigger the whole response, okay? It is inducible, meaning when the cells, we'll get into the cells in a minute, but when the cells actually bind onto a shape, for example, it is now going to be activated. So at that point, you have now induced the immune response. Clonality means once those cells are induced, okay, they are going to make clones of themselves. They're simply going to make multiple copies of themselves to mount up the attack. That's clonality. For unresponsiveness to self, usually these cells involved in adaptive immunity, usually they don't target their own self, right? Usually. Sometimes they do, right? But usually they don't target your cells. They're only supposed to target foreigners, right? Invaders. And lastly, memory, right? So once they have been exposed to a shape, right, like a toxin or a shape on a pathogen, they remember that, okay? They recognize it the next time you're exposed to that same pathogen so that the amount of attack goes more quickly and you don't even get sick. You get sick, why? Because your immune system got caught off guard, right? It didn't recognize the pathogen. But the next time you see it, you don't get sick, makes sense? Because your immune system takes care of it early on and you never get sick, got it? That's the memory part. Now, in terms of the major players, who are the major players in adaptive immunity? They are your lymphocytes. Now, in the last chapter, right, we talked about all our leukocytes, right? And we really didn't talk about them too much instead because they will be in chapter 16. Now, your lymphocytes are basically what? They're leukocytes, right? They are white blood cells. They are the smallest of all the leukocytes, okay? If you see this guy here, it has a really huge nucleus. Very little cytoplasm, okay? And they are the smallest of all the leukocytes. They're lymphocytes. They're going to be involved in what? Adaptive immunity. Now, in terms of the two particular types of lymphocytes we care about in this, are going to be your B cells and your T cells. Okay, why are they named that? First of all, whether they're B cells or B lymphocytes or T cells, T lymphocytes, they both originate from the red bone marrow. Okay, they both start there. They both are, quote, born there. So they both come from bone marrow, period. Now, in terms of where they grow up, in terms of where they actually mature, okay, the B cells stay in the bone marrow, and that's where they grow up. That's where they mature. Now, with these T cells, they start where? In the bone marrow. But then they migrate away from the bone marrow, they go to the thymus gland, and that's where they grow up, that's where they mature, and that's why they're called T cells. Now, under the microscope, they look the same, okay? There's no way to really tell them apart under a light microscope. They look exactly the same. The only way you know whether it's a T cell or a B cell, you look for the proteins on the actual cell. That's it. They have different proteins on them. And that's how you distinguish between B cells and T cells. Now, in terms of the immune response they can mediate, they either do humoral or cell mediation. Now, humoral comes from the Latin word humor, which means fluid or liquid. Now, the humoral immune response, this is the B cell mediated response. You can liken it to the antibody mediated response because B cells produce your antibodies. Okay, so got to get that? Usually, not usually, with the humoral immune response, we're talking about attacking invaders in the blood itself. Okay? So humor is liquid. You attack the invaders in the blood, you make your antibodies, and they target them for destruction. So think of extracellular pathogens here, okay? Extracellular pathogens with the humoral response. Now with the cell-mediated immune response, this is going to be your T cells, okay? Now T cells do not, I repeat, they do not make antibodies. What they do is they look for cells that have intracellular parasites. So cells with viruses, the viruses are where? On the inside, right? They look for cells that have viruses, and they basically target them for destruction. Now, both of the cells, both T cells and B cells, they can also participate in intercellular communications. They can talk to each other, right? They can communicate, they can both do that. But keep in mind, with cell-mediated, it's T cells, right? No antibodies, okay? With humoral, basically, antibody response, B cells. Anything else I forgot to mention? Now with the T cells, they are like the commanders of your army. They actually mediate the whole response itself. They can control themselves as well as B cells. Okay? So think of your B cells as your foot soldiers. Got it? They're your soldiers. Think of the T cells like the commander of the army. They control everybody, right? They send the soldiers out. They tell them what to do. Make sense? Okay.
Excuse me. Which one is the commander of the army? The T cells?
T cells. T cells. B cells are the soldiers. Okay. Now, in terms of your surveillance system, right? The guys who with the scopes, right? The scopes, and they're looking for the invaders. This is the surveillance, right? Like ADT. But it does not cost you $99 a month, okay? It's free. It's free, okay? The surveillance system, in this case, is going to be the lymphatic system. That's the job. They look out for invaders. They are the guys on the fence, right, with the scopes, looking out for who's coming, right?
Are they snipers as well?
What's that?
Are they snipers?
Yeah! Yeah, they got snipers too. They're good, right? Now, within your lymphatic system, basically, they're doing what? Surveillance, right? They're screening fluids for pathogens, okay? Within that lymphatic system, you got the lymphatic vessels, right? The actual fluid, and actual tissues and cells and organs. We'll talk about all of them today. Let's talk about the lymphatic vessels and the lymphatic fluid. Now, the lymphatic vessels, okay, they travel one way essentially. So simply put, they carry lymph. Now, lymph is basically like blood plasma in terms of it's colorless, it's watery, it's just a fluid. Lymph actually comes from the blood. And essentially, you have blood vessels, right? Some fluid leaks out of the blood vessel, leaks into the neighboring tissues, and then gets into the lymph vessels. Now, the lymph vessels, all of them travel through lymph nodes. I'll show you that in a minute. And then the lymph vessels basically travel back to the heart. That's it. That's how it works. So you got lymph vessels, right? They carry all the lymph through lymph nodes where you do the filtering. And the fluid makes its way back to the heart. Anything else here? Now the lymph, okay, carries waste products. So it's not like blood, right? Blood carries nutrients, okay? Lymph carries waste products like broken down proteins or even toxins, okay? It's like you're flushing the toilet essentially. So it's basically waste products carried in the lymph. Now since you've got waste products in the lymph, that's why you got to filter it out, right? Where do you filter it? Ouch, stupid table. Where do you filter it? Through your lymph nodes. Now you already know, you've been sick a few times in your life. You already know where they are, right? Like in your neck, right? You get sick, you feel that stuff right there? Lymph nodes swelling up, right? Armpit, right? Under your armpit. My clicker's dying, my pointer's dying. Lots of lymph nodes there, right? They're filtering out the pathogens. Also, you don't want to say it right, but go ahead and say it in the groin, okay? They're in the groin too, so you've got infection around there. They will also swell up, okay? Now, I will talk about in a minute the other organs that are involved in the lymphatic system, but I'll talk about them in a minute. What I want you to focus on is this actual lymph node. It looks like a little bean, sort of almost like a kidney almost, but it's the lymph node, okay? Now, the lymph is going to come in through the lymph node, through lots of vessels, okay? Don't worry about the name, but lots of vessels. Let's use this meter stick. Yeah, old school. See all these vessels here? They are going to bring in that lymph, okay? Just like one-way flow. Now, as the lymph travels through this lymph node, it's going to make its way out. Now, in this area here, that medulla region, don't worry about the name. Just know in here is where you got B cells sitting and waiting, okay? That's where the surveillance takes place. Those are where your snipers are. The snipers are in this region here, the medulla region, waiting right there, because the lymph has to go through there, right, to get out. Makes sense? Right? Has to go through here to get out. So you got B cells waiting for something to come through. And let's say they find something like some pathogen coming through there. Well, they're going to sound the alarm, right? Uh-oh, someone's in here, right? Let's get to work. Well, in this region here, in this cortex region, you also have B cells, but at this point, what they're going to do, they're going to start to do what? Multiplying, right? Proliferating like crazy. So in other words, if they're multiplying like crazy, your lymph node will probably do what? Probably what? Swell, right? So the only reason why when you get sick sometimes, and you start to feel those lymph nodes, that's the B cells starting to replicate like crazy, and the lymph node actually gets bigger. Makes sense? Because they're trying to do what? Mount the attack, right? Someone is here, we got to take them out. Makes sense? Simple as that. Now let's talk about the other organs involved in this lymphatic system, right? Talked about the lymph vessels, right? Carry the lymph, the fluid, where? Through the lymph nodes where we filter it out, correct? There's some other organs helping us out here, okay? Now we've got some primary organs, okay? The primary organs, why do you think they're called primary? What did I say about bone marrow and thymus? What did I say about them?
That's where the cells mature.
Exactly, okay. That's where the B cells and the T cells actually grow up. They mature there. So they are the primary organs in the lymphatic system. And now you got these secondary organs, right? These guys actually help out with the surveillance. They actually help out with the filtering process, okay? And you've got, of course, we already talked about lymph nodes, right? Lymph nodes, spleen, tonsils, and something called malt. And this is not malt liquor, okay? It's not malt liquor. Not that. MALT stands for mucosa-associated lymphatic tissue. Basically, think about your mucous membranes. Think about mucous membranes like respiratory tract, okay, intestinal tract, all those things. And that's where you have lymphatic tissue. So let me back up and show you where all the MALT guys are. For example, these guys here within that small intestine, all these little patches there, they're called Peyer's patches, okay? They're simply lymphatic tissue within the intestinal tract. Their job is basically to filter out, look for pathogens, okay? So any type of lymphatic tissue within the mucous membranes, okay, they're going to be called the MALT. Got it? MALT. Now, most of your lymphocytes, are actually in the MALT. That's where they are. So primarily the B cells, because I'll tell you right now, the next slide, B cells are not floating around in the blood, okay? They're not doing that. They're somewhere, like in the lymph node. But most of your B cells, they're hanging out in the MALT. Now, both tonsils and the MALT, they primarily capture the pathogens. That's why, for example, you gotta get your tonsils taken out because they've caught so many pathogens it's now infected, makes sense? So you just get rid of it. So primarily the tonsils and the mold, they basically capture pathogens, okay? And most of the B cells are in the mold. That's where they hang out. Okay. Now, let's talk about the bad guys, okay? We talked about the surveillance system, right? The snipers. Let's talk about the invaders, right? The bad guys. They are basically antigens. So an antigen is basically anything that can bind to a B cell or a T cell and trigger an immune response. That is it. Okay? So an antigen is basically anything like pollen or dander, okay, or a pathogen that can bind a B cell or a T cell and actually trigger an immune response. Now, in terms of what the B cells or T cells are binding onto, know this word, they're actually binding onto, they're actually recognizing and then binding onto something called an epitope. So an epitope is on the antigen, okay? So far, an antigen is something foreign, basically, it's foreign. Now, on that foreign thing, it can be pollen or bacterium. Within that antigen, you have a 3D shape. It can be any shape, okay? You have a three-dimensional shape called an epitope. Now, that epitope is what the B cell or the T cell is actually binding onto, okay? That's it. It's the epitope. Get it?
If it binds to one thing, does it deactivate all the other binding sites, or can one T or B cell bind to multiple antigens?
Oh, absolutely.
Okay.
We'll get into that today. So in other words, B cells, well, I'll say that. I'll answer your question. That's going to be today.
Okay.
Absolutely.
There are different types.
Now, real quick, what are epitopes? Okay, epitopes can be anything, okay? It can be various parts of the bacterial cell wall. It can be parts of the flagellum. It can be parts of pili. I mean, you name it, it can be it. It can be pollen, dander. You name it, it can be epitopes, okay? So anything that's three-dimensional that can actually bind to a B cell or T cell to activate it, okay? Epitopes. I just mentioned already, right? Food particles, right? Food allergies, they can also be epitopes. So some people can't eat peanuts, for example. That's why they eat peanuts. They trigger the immune response, okay? Also, dust, right, can trigger immune responses. I like this thing. This is cool. I like old school. Yeah, hold on, real quick. My battery break, let's see that one. So these guys, you know I'm horrible. I used to teach chemistry. My batteries are corroded, so I already know they're not going to work. I'm just hoping and praying that maybe a miracle would happen. I should know that. Goodness gracious. I actually like that music. I felt like an old school professor. Yeah! Back in action now. Okay, let's back over. Okay. Now, let's go over different types of antigens, okay? Now, here's a little cell. Let's just say this cell doesn't belong. Now, we look on this antigen. So the antigen is basically what can trigger an immune response. So far, so good? That's an antigen. What can trigger an immune response? And within that antigen, you have some three-dimensional shapes. They're three-dimensional shapes that literally, okay, they literally bind to a B cell or a T cell. They are the epitopes, got it? See how they have little triangles and circles and little pentagons? They are epitopes. Epitopes are also known as antigenic determinants because they actually trigger the immune response. In other words, if a B cell were to bind onto an epitope, it's activated. Makes sense? If a T cell were to bind an epitope, guess what? It is activated. It's turned on. Okay. Now let's go into different kinds of antigens, okay? You've got exogenous antigens. Exo means outside, right? Like exit. Now an exogenous antigen is any antigen outside of the cell, right? It could be like a bacterial cell. It could be a toxin, right? It could be a virus. Anything on the outside of the cell that can trigger what? An immune response. It's said to be what? Exogenous. So here's a little guy here. He can be an antigen. Within this bacterial cell, see his little flagellum right here? And he has proteins on his flagellum. They can also be what? Exogenous antigens. As long as it's not in your cell, right? Exogenous. Now, endogenous antigens. Endo means what? Within, right? Within. These are antigens that start on the inside of the cell, but they have to be presented on the outside of the cell to be recognized. We'll talk about that on Tuesday. In other words, how do you present antigens that start on the inside? How do you present them where? On the outside to be recognized. So let's say this guy is a virus, right? A virus tend to what, go inside. Well, the viral antigens will be presented on the outside of that cell, right? That cell says, help me, right? I have a virus. Here's the proof, right? It's literally going to take some of the viral proteins, put it on the outside so that somebody can come and recognize it. So if that's the case, if the antigen starts on the inside and it's presented on the outside, that's what? Endogenous. And lastly, we've got auto, meaning self-antigens. Now here's a normal cell. This is your cell, right? And every cell has proteins on the outside, makes sense? So they can be antigens. Now if you have immune cells, if you have B cells or even T cells that are recognizing autoantigens, they have to be destroyed. You know why? Because if they're not destroyed, they're going to start attacking your cells, makes sense? You don't want that to happen. So any B cell or any T cell that is actually recognizing your own antigens that are actually on your own cells, they have to be taken out. They have gone rogue, okay? Take them out of the army. Take them out. Discharge them. All right? Okay. Now let's talk about our G.I. Joe guys here. Our soldiers, right? The soldiers of the army. We talked about the villains, right? Here are our soldiers, the B cells. And these are the guys that actually produce those antibodies. Okay, real quick. We talked about where they start, where they mature, right? Basic training and everything. What? Bone marrow. Found primarily in the malt. You can find them in the spleen, yes, in the lymph nodes, right? Because in the lymph nodes are doing what? Surveillance. Primarily, they're in the malts. Only a very small percent of them are actually floating around in the blood, okay? They're somewhere else, they're in some tissue. But their main job is what I just bolded for you. Their main function is to produce and secrete antibodies. That's their job. They're the foot soldiers. Now, before I go into more words, I want to show you why are B cells so doggone specific, right? Because if this is adaptive immunity, we have to talk about how do B cells recognize specific epitopes, right? So let's do that. Now here is part of the B cell. We're only focusing on his membrane, see that? So the rest of the cell is way down there somewhere. We're focusing on the membrane, and on the membrane, we've got this thing right here. It's called a B cell receptor, or BCR for short. That you should absolutely know, okay? So within the membrane of all B cells, okay, I repeat, within the membrane of all B cells, you've got B cell receptors, BCR. Okay. They look like Y's, right here it is. It looks like a Y. Now they're made up of basically two heavy chains, right? The heavy chains are the big long ones, right? You've got two heavy chains and then two light chains, and they actually form a Y shape. And it's actually embedded in the membrane. So they're not coming off, okay? They're stuck. They're stuck there. They're not coming off. They have another set of proteins that help to keep them in the membrane. All right? Now, let's look at their arms here. Their arms, at the ends of their arms, they got hands, right? I am that thing, okay? Here are my arms, here are my hands, okay? At the end of their arms, they've got hands. Their hands are the variable regions. Okay? That's basically where they recognize and bind something very specific. Got it? So each hand recognizes only one thing, okay? And one thing only. See that? So one hand, one variable region will recognize and bind onto one thing. Period. Period. One thing only. So far, so good? Now, those hands, they're called what? Antigen binding sites, right? Because that's where they bind what? The antigen. Actually, they bind the epitope on the antigen. Make sense? See this guy here? So this big thing is the antigen. There's a little epitope, right? That little shape. And it's going to bind right in there. It's like fitting a piece to a puzzle. That's how it works, right? It's like fitting a piece onto a puzzle. They fit perfectly. And when they come together, it triggers, it activates that B cell. Make sense? It makes them turn on, and he's activated. He's going to do all kinds of cool things, okay? So far, so good? Okay. So now let's get into the words. Now that you have a picture of what a BCR is, right? BCR, B cell receptor. This is where we get the specificity, right? Each hand recognizes one thing and one thing only. All right, so let's get into our words. Blah, 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 blah. Here's the word, okay. Those BCRs are basically called an immunoglobulin. Immuno means like immune system, right? Globulin refers to protein. So though that Y-shaped guy, that's a protein, okay? It is a protein. So that BCR is referred to as an immunoglobulin. Okay, immuno means what? Immune system specific. Globulin just means a protein, period. Now each B cell has more than multiple copies. Each B cell can have up to 500,000 BCRs on the surface of it. So far so good. So each B cell can have up to 500,000 Ys on the entire cell. Main job, look for something, right? Hold on to something. Now here's the deal. Even though one B cell can have 500,000 BCRs, all of those BCRs will only recognize one thing. Make sense? All right? So even though one B cell has 500,000 BCRs, all 500,000 BCRs have the same pair of hands, okay? Which means they only bind to one thing. Got it? Which means different B cells will bind to what? Different things. Make sense? They're like little snowflakes, right? Snowflakes, there's no two that are the same, right? There are no two B cells that are the same. Make sense? So every B cell has its own what? BCR. Make sense? That's why, so going back to your question, Andrew, you talked about, let's say you have a bacterium in your system, and it has multiple epitopes, it has multiple sites, right, like flagellum, or maybe pili, or maybe the capsule itself. You've got multiple, hear me out, you've got multiple epitopes on a given antigen, makes sense? Which means you can have multiple B cells with their own set of hands, right? And they can bind to all the different epitopes on your antigen so that that pathogen does not escape surveillance, makes sense? That's okay, it's like a backup on top of a backup on top of a backup system, right? Put it to you this way, if you can't catch it, okay? Do it again with math. You got a bacterial cell, right? And let's say you got 10 different epitopes, basically what? 10 different shapes on that bacterial cell. Well, guess what? You've got B cells that will match up to those 10 different shapes, just so that that bacterial cell does not escape surveillance. Make sense? So every B cell has its own set of hands, right? And that hand will recognize one shape. Got it? So that's how they back each other up. Okay? That's how it works. Right? So I talked about that, right? That basically says what? A given B cell makes one kind of BCR, which means this, right? One kind of BCR has one set of hands, which only recognizes what? One thing. Blah, blah, blah. The two variable regions are basically the hands, right? That's that. I've spelled that out about 10 times. Each BCR recognizes what? One thing. But you have multiple BCRs, right? Every B cell, just like a snowflake, every B cell has its own set of hands, which means they recognize up to 1 billion different things, literally. Up to 1 billion pairs of hands looking for something to bind, okay? That's how it works. Now, antibodies, we'll skip this line here, okay? And I'll come back to this out of order. It doesn't make sense, okay? Now, let's say, for example, let's say your pair of hands on your BCR, right? B cell receptor, right? Let's say your hands actually bound onto something. And I said it's going to do what? Activate that B cell, makes sense? So what does that even mean? When I say activate that B cell will now start to make clones of itself. Okay, that's what it means. When that B cell is activated, it makes clones of itself, and all of the clones, they have the same set of BCRs, right? Right? All the clones have the same pair of hands, but now they actually secrete the BCR. So let me back up. Okay, so right now, he's about to be what? Activated, right? Correct? Because he's about to bind onto something, he's going to be turned on. If he's turned on, he's going to make clones of himself. Remember that clonality part I mentioned the very first slide? He's going to make clones of himself. Now, all of the clones will have the same Y, right? Correct? All the clones will have the same Y, which means they're going to recognize the same epitope, correct? But here's the deal. All the clones will actually secrete out their BCR. See how this guy is stationary? Yeah. He's not going anywhere. The clones release the BCRs. Got it? Here's the deal. A released BCR is your antibody. Got it? So a released
BCR.
BCR. From all those clones, they are your antibodies, okay? Now the cell that actually did that, the cell that was actually the clone guy, and he made his own BCRs, and he let them go, okay? They're called plasma cells. So plasma cells are basically what turned on or activated B cells that literally release their BCRs, but at that point, if they're released, they're called antibodies. And basically, they have the same shape as the BCR. They look like Ys, makes sense, with little hands that bind onto something. The only difference is that they're missing this thing, right? Because if they still have that, they're going to be stuck, makes sense? So antibodies look just like this, except they don't have that, and they are free. They're in your blood looking for something, correct? Blah, 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 blah. Last part is very important, right? The antibodies, right, or the BCRs, are going to be the same as the actual activated B cell, right? They're all the same. Make sense? They're all the same. So the antibodies that got released, they're basically the same as the very first guy, right, who started the whole process out, correct? All right, good. It makes sense, right? It's not that difficult. Just stay with me. Basically, that's your antibody, right? See how he's free? He's free. That's your antibody. Now, notice we got some parts labeled. These are important parts here. We still got our Y, right? We still have the variable region. So I said the variable region is basically the hands at the end of your arm. Makes sense? You still have that, but now they're labeled. Pay attention to this stem here. The stem at this point, when it's released, okay, has a name. The stem is called the FC region. This is going to be really important, okay? So that stem on your antibody is called the FC region. Basically means fragment. F is fragment. C just means you can crystallize out a solution, but that's not important. Just know right now that that stem is the FC region. Got it? That will come up later on. Now, what happens when your antibody binds onto an epitope? You got several different results, okay? Well, first, we can activate our complement system. Was that the last chapter? The whole cascade, and then with the match tap, okay? You can activate the complement system when antibodies bind onto something. You can even trigger the inflammatory response. That's really part of the second line of defense, okay? And all these guys, I'll talk about in pictures because I like pictures. We'll talk about neutralization, opsonization, killing by oxidation, all these Asians, agglutination, and then antibody-dependent cellular cytotoxicity, or ADCC for short, okay? That's going to be important. ADCC for short. Now let's go to the pictures, and we'll talk about each one in detail in terms of how it works. Now, next test, we're all on this stuff, and it's not easy stuff. Okay? You should already know how the complement system works, right? That's why it's not up here. That was the last chapter. You should already know how inflammation works, right? Last chapter. Let's do the new guys, okay? Neutralization. This is where you have some antibodies, and they're going to either A, cover a toxin, okay, here's a toxin, he's covered, right? Or they are going to bind to adhesion or adhesive molecules found on bacteria or on a virus. Now, what are the adhesion molecules or the adhesion proteins? What are they involved in?
Benson?
What's that? Attachment, right? So adhesins help with attachment. I believe in some chapter I said in order for a pathogen to make you sick, they first of all have to get into you, right? But they have to literally, it's not an option, they have to attach to your cell. If they can't attach to your cell, they can't make you sick, right? So that's why in neutralization, you've got antibodies, and they can coat the adhesion proteins on bacteria or viruses so that they can no longer do just that. They can adhere to your cells, which means they can make you sick. Now, if they cover a toxin, they literally neutralize the toxin so that the toxin cannot make you sick. Okay? And opsonization, this is like magnified phagocytosis simply, so it's like amplified or magnified. Yeah, opsonization, phagocytosis. Yeah. So in opsonization, literally, here is a pseudopod from a phagocyte. It can be from a neutrophil or a macrophage. Okay, they are phagocytes. And within the pseudopod, it's going to actually bind onto the FC region of some antibody. Okay. So right now, within that pseudopod, it's like a little, like this, right, like a little cup. it's got some antibodies waiting, right? Waiting to grab onto something. Now let's say some poor bacterium finds its way in that area, right? And the antibodies will lock onto him, okay? See how this poor guy is stuck? See that? The antibodies have locked onto him. These antibodies are within the phagocyte. Well, now when that happens, the pseudopod will now close in on him, okay? It's going to eat him up. So opsonization, you see how that guy is covered in antibodies? It triggers phagocytosis. Even though nothing else, opsonization triggers phagocytosis. Killing by oxidation, these antibodies can actually catalyze reactions. In other words, they can actually produce hydrogen peroxide, which we know is bad for cells, right? It's bad for your cells, bad for bacterial cells, okay? So sometimes antibodies, they can bind onto antigens and epitopes, and they can actually start to produce hydrogen peroxide, which is toxic, correct? So that's death by oxidation. Now, here we go. Agglutination. So in agglutination, you have numerous antibodies binding onto your antigen. And when that happens, they literally clump together. So that word agglutination means the clumping together of something because of the antibodies making them clump together, right? And if these guys are clumped together, they're not free to make you sick, make sense? They're stuck. So that's what agglutination. When antibodies literally coat your antigens like these bacteria, and they literally clump together, which means they can't do anything. They're kind of stuck, all right? They're done. Now, the last one, the ADCC, antibody-dependent cellular cytotoxicity. Ooh, and I know it's a tough one to say. Here we've got an NK lymphocyte, that natural killer guy. That was in the last chapter. Now, he can also bind to an antibody with that same FC region. Basically, FC is the stem, right? That's all that is. FC is the stem part of the Y. Now, when the hands actually bind onto this poor guy, it's going to activate that NK cell. So when he's activated, he makes two types of proteins, okay? The first protein is kind of small. It's called perforin. Perforin makes holes. Simply put, perforin perforates, right? Perforin makes holes in the membrane of that poor cell, okay? The next protein is going to be granzyme. Granzyme. Now, granzyme goes in, okay? First, you got to make a hole, right? Make a hole. Then granzyme, it comes from the NK. It comes from the NK cell. He does all of that. Then you got a hole. Now, granzyme goes in, and granzyme actually triggers cell suicide, or apoptosis. So this poor guy here, he's going to basically kill himself. In other words, the NK guy's not going to do it. He's going to make that poor cell kill itself by apoptosis, okay? So whenever you hear apoptosis, that is literally programmed cell death, cell suicide. So this poor cell is going to kill itself. He's gone. So these are the mechanisms by which antibodies work when they actually do what? Bind onto something, okay? All right. Now at this point, I wish I could go on, but I can't. I have to let you go. So I will see you on Tuesday with more stuff on B cells and T cells. It is.